acervos culturales más importantes de nuestro país.
¿Cómo ha contribuido Miguel León Portilla a la cultura de México? Yo creo que ha hecho él solo, desde sus posiciones políticas, culturales, filosóficas sobre los pueblos originarios, ha logrado él solo más que muchísimas políticas lingüísticas de gobierno. Miguel tiene una obra multidisciplinaria porque su visión de los pueblos es multicultural, como debe ser cualquier pueblo. Miguel ha insistido una y otra vez en que las lenguas son patrimonios intangibles de los seres humanos y que cuando se pierde una lengua, se pierde un patrimonio para la humanidad. También ha hecho Miguel algo que es una reflexión profundísima sobre cómo fue el contacto, el encontronazo de la conquista. Y Miguel encontró, cuando era embajador en la UNESCO, encontró un eufemismo buenísimo, hay que decirlo, que fue encuentro de dos mundos. Me parece que él su impulso desechó un término muy desafortunado para la cultura, que es el descubrimiento de América, y lo llamó con un acertadísimo eufemismo, políticamente muy, muy acertado, y puso las aguas un poquito en calma en el año 92, Encuentro de dos mundos. Miguel tiene obra trascendente y profundísima sobre cómo se dio este encuentro. Y además es un hombre altamente ejecutivo, o sea, si él te dice esto hay que hacerlo y lo vamos a hacer así, Miguel León Portilla lo hace así. O sea, no solamente está en este mundo filosófico, en el discurso defensor de los pueblos indígenas, sino está en la ejecución de cómo hacer más visibles y darles respeto. Yo creo que ha contribuido a quitar una cantidad gigante de estereotipos que caían y recaen, por desgracia, todavía en los pueblos indígenas.
Quisiera destacar un aspecto de la enorme obra de don Miguel León Portilla, que es la que se relaciona con su defensa de los pueblos amerindios y de sus lenguas. Al paso de los años, don Miguel se ha convertido, diría yo, en el principal portavoz de la lucha de los pueblos amerindios y también de muchos lingüistas como yo, porque se respete la dignidad de estos pueblos, se respeten sus costumbres y se respeten sus lenguas. En ese sentido, el papel de don Miguel es invaluable. Donde quiera que uno se interese por saber qué hacer en favor de nuestras lenguas amerindias, siempre va a tener uno que consultar lo que ha escrito don Miguel León Portilla e incluso su actividad permanente. Quizás ahora no por su salud delicada, pero todos lo hemos visto participar activamente en esta promoción de los pueblos y de sus lenguas. Yo creo que es uno de los aspectos de don Miguel que van más allá del filólogo y el historiador que es, a un compromiso con nuestros pueblos amerindios.
En la historia de México, una mujer fue capaz
Para mí y para todos los mexicanos, don Miguel León Portilla debe representar el mejor ejemplo de lo que es un hombre de cultura, un hombre que aprendió tanto como pudo de su entorno, que recogió la cultura pasada y presente de los mexicanos y que nos está dejando una herencia cultural absolutamente extraordinaria. A mí me impresionó cuando, siendo muy chico, estando en el bachillerato, leí La visión de los vencidos, y es una obra epopéyica absolutamente comparable a la Ilíada, una de las grandes tragedias de la historia de la humanidad. Pero probablemente el aspecto que a mí me conmueve más de don Miguel, aparte de tener la suerte de muchas veces sentarme a su lado y de sentirme un amigo de él, es el hecho de que para León Portilla los indígenas no son solamente parte de ese pasado excepcional que tuvimos, capaz de hacer pirámides o estatuas y piezas de jade y obsidiana, sino que para don Miguel León Portilla también los indígenas son ese grupo enorme de mexicanos que hemos marginado, que tienen una riqueza extraordinaria en términos culturales, en términos de conocimiento del ambiente, en términos lingüísticos, y para él no son solamente un vestigio del pasado, sino una realidad viva de México. Yo creo que uno de los grandes regalos que a mí me ha hecho el Colegio Nacional es la posibilidad de conocer y tratar a don Miguel y de haber recibido de mano suya La visión de los vencidos en náhuatl, que me parece la mejor demostración del compromiso de la cultura mexicana contemporánea con nuestro pasado y con ese otro componente del México que él se ha encargado de recuperar y llevar hacia la línea de primera.
Hola, soy Mardonio Caraballo, soy escritor, escribo en náhuatl y en castellano, Nixtlapaloa, Nochi Noixnimen, Nopatlentle Machtiani, tocan Miguel León Portilla. Hola, ¿cómo están? Bueno, envío un saludo al maestro Miguel León Portilla. Pienso que la obra del maestro ha sido fundamental para entender a México, sobre todo desde sus personas y sus habitantes anteriores, los que llegaron primero, como dirían los zapatistas, ¿no? Pienso que es fundamental su entendimiento para con aquellos hombres y mujeres de los cuales nos había contado una versión que en algún sentido dignificara, a su vez que acusara todo lo que había ocurrido en un tiempo. Creo que es importante, es un hombre de su tiempo, si bien pienso que la gran contribución de Miguel León Portilla sobrevive al tiempo, también pienso que ha acuñado de cuya agua han abrevado múltiples artistas, múltiples escritores, ahora en lenguas originarias, que sin duda están haciendo una revolución con respecto de quiénes son, con respecto de la identidad, con respecto de México, quiénes somos los mexicanos, por qué somos como somos y por qué nos vemos como nos vemos. Y pienso que todas esas reflexiones no podrían haberse dado si no hubiese un inicio, y pienso que en ese sentido, en la historia, para entender la historia que tenemos ahora, para comprender a los pueblos indígenas en la narrativa nacional, me parece fundamental el agua, digamos, el abrevadero que se llama Miguel León Portilla.
Hola, ¿qué tal? Soy Juan Villoro, escritor y periodista, y me da mucho gusto poder hacer una evocación de Miguel León Portilla, uno de los grandes intelectuales de México, que ha sido un intercesor extraordinario entre la cultura y la lengua de los pueblos originarios, principalmente del náhuatl, y nosotros. Miguel León Portilla ha sido ese intermediario esencial entre un mundo desaparecido, derrotado, de los primeros mexicanos, que es el mundo de la visión de los vencidos, pero que todavía perdura entre nosotros, perdura sin una voz propia que se escuche y que gracias a intercesores como Miguel León Portilla puede ser reconocido. Entonces ha sido valiosísimo este trabajo de entender la importancia, el significado del legado indígena. Ahora bien, creo que el gran legado de León Portilla tiene que ver con el pasado, pero también con el futuro. Él apunta a una cultura que ha sido sojuzgada, destruida, pero que puede ser todavía rescatada y puede perdurar entre nosotros, pero creo que implícitamente también está señalando a un futuro. ¿Cómo seremos los mexicanos del porvenir? Y yo creo que ahí esta figura puente nos está diciendo la verdadera historia de los pueblos originarios será escrita en su lengua o en sus muchas lenguas y será escrita por ellos mismos. Llegará un momento en que México no tendrá necesidad de intermediarios, de intercesores para conocer esa cultura, porque esa cultura volverá a estar viva entre nosotros. De modo que creo que el gran legado de Miguel León Portilla tiene que ver con la recuperación de un pasado, pero también con el señalamiento de que hay una aventura por venir, que es la de que los propios pueblos escriban su historia en la lengua que él ha traído hasta nosotros.
Buenas tardes, a nombre del Colegio Nacional y en particular del doctor Sarucán, del mío propio, les queremos dar la bienvenida a este viernes de la evolución, correspondiente a septiembre, en donde, como todos ustedes saben, recibieron unos anteojitos de estos como para ver películas del santo o contra las momias o cosas así. La razón de que reciban ustedes estos anteojitos es muy simple. Hoy tenemos el placer de tener a uno de los grandes expertos en la estructura y la evolución del ribosoma, que es el doctor Anton Petrov de Georgia Tech, en Atlanta, en Estados Unidos. Él es un amigo muy querido, es una persona muy cercana al Laboratorio de Origen de la Vida de la Facultad de Ciencias y él ha desarrollado, junto con el grupo de expertos de origen y evolución del ribosoma que se encuentran en Atlanta, ha desarrollado los modelos más refinados que tenemos, los modelos tridimensionales más refinados que tenemos del ribosoma. Y yo he tenido la suerte de estar en su cubículo, en su oficina, que se puede oscurecer por completo, y con goggles especiales uno ve realmente flotar en el espacio el ribosoma y sus componentes. Después de que invitamos al doctor Petrov, hubo la discusión de cuáles eran la mejor manera de lograr el mismo efecto en la charla de aquí, y él muy amablemente estuvo ensayando distintas opciones, y los anteojitos estos que tienen ustedes van a ver que es lo más adecuado. ¿Por qué nos interesa el ribosoma? Bueno, porque es una de las estructuras más conservadas y más antiguas que podemos reconocer en todas las células que existen, en todos los organismos. Nos interesa porque de esa manera, estudiándolo, podemos comprender el proceso de síntesis de proteínas, que es un proceso esencial para todas las formas de vida que conocemos. Nos interesa porque la exactitud con la que el ribosoma traduce el mensaje genético va a generar proteínas que pueden ser o no defectuosas y que pueden tener o no efectos graves, severos en la supervivencia de las células, y porque, ahora lo sabemos muy bien, fenómenos como el efecto de antibióticos, el efecto de una serie de medicamentos se da directamente sobre el ribosoma. Es decir, lo que el doctor Petrov hace es estudiar una de las estructuras más antiguas, mejor conservadas, que existen en toda la biosfera, Y ese conocimiento tiene no solamente la riqueza de la comprensión teórica de un fenómeno como el de la evolución, sino también una serie de implicaciones prácticas, médicas, sociales, extraordinariamente concretas, sobre todo ahora que vemos y sufrimos el fenómeno de la resistencia a los antibióticos. Estos estudios demuestran lo absurdo que es pretender separar ciencia básica de ciencia con aplicación, porque se dan ustedes cuenta que lo que la ciencia produce es conocimiento que nos puede servir en situaciones muy específicas. El doctor Petrov estudió en Perm, la ciudad rusa que los que hayan leído el doctor Zhivago, tienen ahí, es un sitio muy importante en los montes Urales, en la región de los Urales de la vida rusa, de la vida cultural, de la vida científica. Él estudió física y química, hizo una maestría en físico-química y después ya su doctorado estuvo dedicado específicamente a problemas de biología celular e interacción con fagos. Ese postdoctorado lo hizo en Georgia Tech y allí conoció al doctor Loren Williams, que hemos tenido aquí en el Colegio Nacional, un conferencista también extraordinario, y encontró su verdadera vocación, que es efectivamente el estudio tridimensional de sistemas macromoleculares celulares. Algunas de las partes de la charla, sobre todo cuando veamos en tercera dimensión el ribosoma creciendo, evolucionando, moviéndose, tienen un fondo musical. Ese fondo musical van a reconocer que es la fuga número 1 de Juan Sebastián Bach, y eso lo tocó el fondo musical en un órgano de Atlanta, la maestra Yelena Dubrilla, esposa del doctor Petrov, de manera que ella es coautora en parte de lo que vamos a oír hoy. Anton, welcome so much. Thank you so much for being with us. And the mic and the audience are yours.
Buenas noches. Hola. And I would like to thank Dr. Antonio Lazcano for inviting me here, and I would like to thank all of you for being here tonight and listening to this story. And this is how we're going to do it tonight. So this presentation will include a number of slides that are projected in a 3D. So most of the slides will have a white background, and those slides, regular slides, you don't need to have those glasses to see the slides in a white background. And once you see the slides with a black background, those are the special slides. Those are done in 3D, in stereo 3D. So once you see black background, please put your glasses on. So without any further ado, I will continue to my presentation. Oops. Let's see. Screen. Okay. Where do we come from? What are we? Where are we going? Those questions that were beautifully posed by Paul Gauguin in 19th century have been asked by humankind for many, many, many centuries before. But it was not until 20th century when scientists, and some of them are listed here, they just started to address those questions scientifically. And it was not until 1967 when Professor Crick from Cambridge formulated what we know as a central dogma of biology. And this central dogma simply suggests that in all biology, in all living organisms, there is a very common pathway to transfer information and to make proteins, which are known as the workhorses of biology. So the central dogma, in other words, it's basically an operating machinery for life. So, and according to the central dogma, all the information in all living species, we know it's stored in the genes as a DNA, and at some point this information is transcribed from DNA to another molecule known as mRNA. And then this information, it is translated into proteins by means of genetic code. And the translation is performed by a special machine, which is called a ribosome. A ribosome is a universal translator, or, if you will, a universal compiler of life. It translates all the information stored in the genes into protein sequences. So, in other words, to understand the question, where the life came from, we can reformulate this question and simply ask, where did ribosome come from? And surprisingly, it was another few years after Professor Crick formulated his central dogma. And at that point, quite a young scientist, Carl Woese, sent a letter to Crick. At that point, there was no emails, so he had to write a letter. And in that letter, a young assistant professor from University Urbana-Champaign was basically asking for his encouragement to pursue and do his work, do his research in a field of ribosomal evolution. And that's what he said. If we are ever to unravel the course of events leading to evolution of prokaryotic or simplest cells, this can be done by using the cell's internal fossil record. The obvious choice of molecules here lies in the components of the translational apparatus. So that was 1971 or 1972. And it was a beautiful prediction or foresight of Carl Woese, because a few years later, after he wrote this email, he and his postdoc at that time, George Fox, discovered a completely something that had not been known before. And that discovery was made by using ribosomal RNA. Specifically, George Fox and Carl Woese have been able to demonstrate that there is an additional domain of life which is known today as archaea. And before this discovery, it was quite clear that every single life was divided into two major categories, prokaryotes and eukaryotes, by simply looking at the microscope and figuring out whether there is a nucleus or no nucleus. And by analyzing ribosomal sequences, Carl Woese and George Fox were able to demonstrate that there is a certain group of prokaryotes are quite different from other prokaryotes known as bacteria. And they call this group archaea. And it appears that those archaea are much closer related to eukarya than any other bacteria. So that turned out to be a quite powerful method, and indeed, the prediction just opened up the entire field known as evolutionary genomics. And all this was done using ribosomal RNA as evolutionary markers. So let me briefly talk about ribosome in a very basic way. Ribosome, it's a macromolecular complex containing 2 RNA molecules and a number of proteins, and consisting of 2 subunits, the large subunit and the small subunit. The large subunit, and those 2 subunits perform different functions. The large subunit is responsible for production of peptide bonds and protein synthesis. The large subunit contains 23S ribosomal RNA and about 35 proteins. A small ribosomal subunit binds the mRNA or messenger RNA, and it also binds another molecules called tRNA that I'm going to talk about in the future. And it's responsible for decoding, meaning reading the information from messenger RNA and converting it to specific protein sequences. So those two subunits, although they are very similar, they perform completely distinct functions. And our understanding of how ribosome works was completely improved by the help of those individuals. Venki Ramakrishnan from Cambridge, late Tom Stites from Yale, and Ada Yonas from Israel. And what they did about 20 years ago, they were able to crystallize ribosome and determine their three-dimensional structure. So now we know not only all the sequences, but we know how ribosome is organized in a three-dimensional space. And it appears, so this is a structure of ribosome. It appears to be quite complicated. It's a macromolecular assembly of 2 million Daltons. So it's very difficult to comprehend the structure of this molecule. So sometimes for simplicity, I will be using what is shown here on the right. Those are secondary structures of ribosomal RNA, the large subunit and the small subunits. So those are two-dimensional projections of ribosomal RNA onto planes. And we can, using those projections, we can basically, similar to Google Maps, we can map all different information so we can easily comprehend it. So let's take a close look and try to summarize everything what I've said so far. So let's start and just take a look on how ribosome is organized in 3D. So we have this mRNA molecule. It's a short fragment of mRNA. It's much larger, but this fragment binds within ribosome. So, and then we have this large subunit where you have in purple ribosomal RNA, and there are a bunch of about 20 ribosomal proteins in green, and this is mRNA right here. And then what we also have, we have a tRNA molecule which binds to an mRNA molecule here at the interface with a small subunit. And it has a tRNA molecule is L-shaped molecule, and it has at this point a free end right here, which carries an amino acid. And then finally, we have a large subunit right here. A large subunit is a part of the ribosome which is responsible for synthesis of peptide bonds, production of the peptide bonds. So, and that's another representation of those two subunits. And finally, we have another tRNA molecule right here. So there is one in dark blue, and this second one in gray. So as of now, we have 5 different molecules: mRNA, small subunit, P-side tRNA, large subunit, and A-side tRNA. And once again, I will repeat it. mRNA, small subunit, P-side tRNA, large subunit, and another tRNA molecule. And what I just showed you, in fact, it's a process of initiation of a translation. That's basically the sequence. It's a little bit more complicated because it includes a number of additional auxiliary proteins known as initiation factors, but the sequence of those events describes the initiation of translation quite well. So you just got yourself familiar with ribosome, its structure in 3D, and you basically saw that this molecule is quite complex. So, but at the same time, it's very rich in history, and it contains a lot of information. And what I'm going to argue next, that similar to geological fossils, which are shown here. This is how geobiology works. So people basically can trace down and excavate different fossils and figure out information about evolution. This is how the first evolutionary tree from Ernst Haeckel was constructed, by excavating different fossils and figuring out the relationship between species. The problem is that the oldest biological fossils are dated to 3.2 billions of years ago, and life has originated at 3.8 billions of years ago. So we do not have any geological record for the origin of life here. But luckily, we have a molecular fossil, which is a ribosome. And I'm going to argue here that we can extract the information by looking at the structure, similarly how we can extract, or people have extracted information about past by doing excavation with this pyramid in Chichen Itza. So when it was discovered about a century ago, in 1930s, there was a first discovery of the inner shell, and then a few years ago, at 2015 or so, there was a discovery of another shell. So another analogy, if you wish, we can look at the ribosome, which is, I'm going to argue that it is very similar to organization of trees. And Leonardo da Vinci was the first one who proposed that some information can be excavated from the trees. Trees record history. The rings around the branches of trees that have been sown shows a number of its years, and which years were wetter or drier to more or less thickness. That's basically a proposal of Leonardo da Vinci. And what we will see in a minute, that ribosome also has this organization by shells or by layers. And therefore, in a very similar process, which I will describe in a detailed way, we can extract the information from those shells. Another analogy would be we can peel those onion layers from the ribosome and go back into our past to our roots of the origin of life. So, and let me just try to convince you that we can do it. So these are so far I was telling you that ribosomes, they are very similar. They exist in every cell in every living organisms. And yet, if you look at different ribosomes, at the structures of different ribosomes available today, you would realize that the central portions of ribosomes are very similar, but different ribosomes have different elements located at their surface. And the central portions are very conserved. The central portions of the ribosomes are even more conserved in a three-dimensional space. So on this plot, we do see a superimposition of two ribosomes. One ribosome is from archaea, the second ribosome is from bacteria. And when they are superimposed to each other, you can clearly see no difference except a few elements here at the surface. So structurally, ribosomal structures are very, very conserved. So, and if you look at how ribosomes are organized, bacterial or archaeal ribosomes are the smallest ones. And if we assume that those are the core of all ribosomes, which is the case, then we see in eukaryotic protists, we do see additional elements located at the surface, and we do see additional proteins, which form another layer. And then once we go to mammalian ribosomes, we do see additional elements extending out from a pre-existing surface here. So that's basically a summary of what I just said. And if you look at the size of ribosomal RNA, it's about 2,900 nucleotides, and the size is approximated by a radius of those circles. So the bacterial and archaeal ribosomes have the smallest size. And then once we go to protists, they are substantially larger, and this is S. cerevisiae, which is a fungi ribosome, but it's still here. So we go from 2,900 nucleotides to 3,800 nucleotides on average. And then once we go to metazoans, we do see a progression, drastic increase in size. So for simplicity, I'm not going to talk about entire ribosome, but I will focus on one fragment called Eukaryotic Expansion segment 7, which is a part of the large ribosomal subunit. And this expansion, of course, it doesn't exist in prokaryotic ribosomes. So this is a helix 25. And it increases in size in archaea slightly, from 30 nucleotides to 80 nucleotides. But then if you look to prokaryotic ribosomes, this expansion becomes more complex, it becomes branched and longer. And then if we go to multicellular species and to metazoans, we do see further increase in complexity and size. So, but there is another very interesting observation here, that at each point, here and here, the new component sort of contains the precursor. So if we compare those two, this one, S. cerevisiae, would be contained within Drosophila, et cetera, et cetera. So what does it mean to us that we've noticed that there is a particular mechanism for ribosomal growth, for growth of ribosomal RNA. And this mechanism is called accretion. So ribosome does accrete ribosomal RNA, new ribosomal RNA, on its underlying core without making any perturbation. And this is an experiment. So we are talking about eukaryotic ribosomes here. But the nature has made this beautiful experiment for us. So, and let's take a closer look at those expansion segments. So this is a slightly different expansion segment, but they will see the same motif here. So this is helix 52 from a large ribosomal subunit. And what you see here in blue, this is how this helix looks in bacteria E. coli. And then the same helix 52 with an additional expansion segment 19 shown as a branch. But what you can see here is that there is no any perturbation to the underlying core upon emergence of this new additional expansion segment. The same is another region, helix 38 and expansion segment 12. So basically, we can easily excise those expansion segments and seal those regions back without perturbing anything. So this beautifully demonstrates the accretion mechanism. And how we can do it, because we realize that there are those expansion fingerprints, those regions at which new elements can be excised. So similarly, the same fingerprints and new elements are observed within the small subunit. So basically here it's E. coli, bacteria E. coli, archaea, Pyrococcus furiosus, fungus, and human sapiens. Again, you can see the same progression, you can see the same fingerprints, you can see a nice superimposition suggesting accretion mechanism. So I hope I've convinced you that going on from common core, from bacterial ribosome to all eukaryotes, ribosome has grown by this accretion mechanism. And here we asked ourselves a simple question. If we can work this process forward, starting from the ribosomal common core, which is a bacterial ribosome, can we, in a similar fashion, go backwards and try to dissect the common core itself? we ask this question, do we see similar elements within the ribosomal core? And surprisingly, we did. So, for example, this one of the helices is helix 2, and there is an insertion. This is how it looks in 2D, and the same insertion fingerprint as I showed you a few minutes ago for eukaryotic expansions, but we do see it now within the common core. And this is just one example. And surprisingly, we saw many of those within the common core. So at that point, we just said, hey, we can break those subunits, large and small subunits, into pieces. And we can identify all possible expansion segments. Okay, that was great, but not good enough. So once we broke our ribosome in those pieces of Lego toys, right? So we have this full set of small fragments. What do we do with them? How do we order those? We needed something. We need some additional information. And apparently we have it. So let me do some experiment here. So I'll put this chair, and I'll sit on this chair. So can you tell me what was here before, this chair or me? Chair. How did you know that? Well, it was a little bit dirty experiment, but let me sit on this chair. So what was here before, this chair or me? Chair, the same answer. So how do you know that the chair was before? Yes, I'm dependent on that chair, right? So I need this chair to hold my weight, right? So, and if we look at molecular interactions between different elements within the ribosome, they appear to be different. So there is a particular type of interaction called A minor interactions, adenine minor interactions between adenines, certain adenines that are flipped out, those yellow bases right here. And they interact with minor grooves. So we have those two helices. One helix doesn't have anything, and it just has a minor groove as an acceptor of those interactions. And then there is this helix, which is a donor of those interactions. So it has adenines. So it's a similar example. Can you tell me, where is the chair here? Which helix is older? Which number? Okay, 75 or 68? Which helix is older? 75 is older. Yes, that's absolutely right. And how do we know this? The same example. So helix 75, it's a chair. And helix 68, it has those adenines, which depend on the presence of helix 75. So in order for those adenines to interact with a minor groove, helix 75 must be already there. So, and that basically beautiful idea was brought by those two individuals, Konstantin Bokov and Sergei Steinberg, and they published this beautiful paper about 10 years ago in Nature. And they mapped all those interactions within the large subunit, and those interactions are bidirectional. So we have a donor and acceptor. So donor of A interactions would be red, acceptor would be yellow. So, and what do they see? All donor parts, acceptor parts would be here. And this is, therefore, can be thought as the oldest part of the ribosome, and surprisingly, that was the peptide transfer center. So, and this is exactly the same what we can do with our elements, and we've done so. So basically, we have broken a large subunit, and there are 60 elements. We broke the small subunits, there are about 30 elements, and we can put them in order using the knowledge of those A minor interactions. Let's just, I will just go briefly over this idea, and this central part of the ribosome known as the peptide transfer center. And I will just show you those elements very briefly. The fact that we can see the same insertion fingerprints, the fact that we can see the same A minor interactions, and that immediately suggests that this helix is older, this helix is newer. It's another example and another example, and the same thing, insertion fingerprint and A minor interactions. So we have, that was just the beginning of the story. And now, let me show you the rest. So far you saw the same central peptide transfer center containing first 5 elements. And you can see here a pore in the central part of the peptide transfer center, which is known as the exit tunnel. So this is a hole where the peptides, new peptides formed, will be exiting throughout this pore. And then there are those elements here, P loop and A loop. Those are the regions where tRNA molecules will bind when they deliver amino acids. And this PTC gets elaborated with additional ribosomal RNA, which sort of embraces it and stabilizes. And later on, so now I'm now approximating, I'm not telling you a story for every 60 elements. I'm combining those elements, some of them, into groups of elements, which are designated here as phases. And we just decided to put those into 6 phases. So phase 4 would be in yellow, phase 3 in green, phase 5 here in gray, and then now phase 6 is in dark gray. So those are 6 phases. And once again, those 6 phases shown by different colors. And interestingly enough, each of those phases contribute to the evolution of the tunnel. So I'll stop here. Let me see. Okay. So, and in a similar fashion, we can do the same procedure for the small subunit, and it's a little bit simpler, and for the sake of time, I'm not going to talk about it. But you got an idea that we can break those ribosomal RNAs into those elements, and then based on information about A minor interactions, we can put those elements in order, and then we can combine those ordered elements into 6 groups that we call phases. So, and based on everything what I just described, we can put those elements onto this timeline, and this timeline, in fact, represents our sort of hypothesis how this ribosomal RNA may have evolved. So, and what does ribosome do? Let me ask you this question. I already mentioned it. It makes proteins, right? How does it make protein? It stitches together amino acids, right? So, if you look at the evolution of sewing machines, and if you look at the evolution of ribosome, they're very similar. So, okay, couple of comments so far. I've talked about large and small subunits. Yes, I talked much more about large subunit, but here let me make a few comments about both of them. They undergo through the same evolutionary processes. They do have the same accretions. They do have the same insertion fingerprints. They evolved in a similar fashion. And yet, those 2 subunits are remarkably different. And this difference, of course, stems from the fact that those 2 subunits perform different functions. And some of the differences are listed here. So they're different by architecture of their catalytic sites. They're different by the topology of their termini. They're different by their shape and flexibility. They're different by their domain architecture. So just to elaborate a little bit here. So we have domains 5' and 3' termini. They're separated in a small subunit. They're together in the large subunit. And of course, this is critical for the small subunit. The separation of those domains is a prerequisite for mRNA binding for the small subunit, because you have a single-stranded region right here. It's a Scheindel-Garner sequence responsible for SSU mRNA interactions. Then there is a difference in flexibility. This is from George Fox. He mapped all those flexible regions, or pivot regions, as he calls them, for both large and small subunits. And you can see that there are a lot of flexible regions in the central part of the small subunit. The large subunit only has a few pivot points located within the peripheral regions, phases 5 and 6. Finally, SSU and LSU are different in their shape and architecture. So LSU has a form of those dendrite, so 4 domains, 4 structural domains of the small subunit. They are specially separated. You can see those 4 domains colored in 4 different colors. LSU has 6 domains, structural domains, but those domains are entangled. And the reason why they're entangled, because the large subunit, we just saw it in the movie, what it does by evolution, it just keeps growing this exit tunnel. You saw a progression, that pore, the exit tunnel for the proteins. And in order to build this pore, in order to build that exit tunnel, ribosomal RNA has to entangle itself to keep that pore open. So, and so far, I've only spoke about ribosomal RNA. Haven't talked anything about proteins yet. Well, it's about the time. So, ribosome contains some proteins that are known to be universal, which means that they're the same in archaea, bacteria, and eukarya. There are some other proteins, they are somewhat more specific to bacteria and archaea. So for purpose of my presentation, I'm only going to focus on universal proteins. So, and again, please put your glasses on and let's take a closer look. This is a particular protein, UL3. And we start with a region that interacts with phase 3 of ribosomal RNA, and this region is located deep inside within the ribosome. And this region doesn't possess any elements of secondary structure or tertiary structure. It's just a random coil. And then once we go a little bit farther out, from phase 3 to phase 4, we start seeing formation of some secondary structure. This is beta hairpin. And then once we move to phase 5, we finally see a three-dimensional fold, one of the simplest folds known as a beta barrel. And if we go to phase 6, which is at the surface of ribosome, we do see a very complicated protein fold that contains beta strands and alpha helices. So once again, we can go through this and see a progression of events. No secondary structures, elements of the secondary structure, simple three-dimensional folds, complicated three-dimensional folds. So basically, we do see a history of a protein fold on a ribosomal protein imprinted within the ribosome. That's a different rendering of the same protein. So, and here is a few more examples. We don't see here all the phases, but this is a ribosomal RNA. Now, these are ribosomal proteins, colored in the same way. You can see that inside the ribosome, there are a lot of unstructured motifs. And as we go inside out, we do see more and more structure with very sophisticated structured proteins located at the surface of ribosome. So ribosome apparently not only make the proteins, but it also learned, taught proteins to fold. And this is our sort of idea how we think of this protein folding process. So we can go from very unstructured to some structured to basic three-dimensional folds to very complicated three-dimensional folds. And all this information is recorded within the ribosome. So, and now, if we combine those two ideas that I've discussed, we have an evolution of ribosomal RNA. We have an evolution of protein folds. And apparently, those two exercises have been done independently from each other. But altogether, there is a beautiful correlation between those two different stories, the story of ribosomal RNA and story of protein folds. So we are arguing that accretion model of ribosomal evolution suggests a co-evolution of rRNA and R proteins. And proteins have learned how to fold at the surface of ribosomes. And the ribosome is essentially, literally, a birthplace of proteins in many respects. So, and if we go from common core, and I already mentioned it earlier on today, and if we go to more sophisticated species, archaea and eukarya, we would see those additional expansion segments all over. So ribosome did not stop growing at the common core once the life emerged. It kept going by adding those new elements, by accreting additional RNA, by accreting additional proteins. And this slide demonstrates the ribosome of all of us, humans. And surprisingly, this is the largest ribosome. A large subunit of this ribosome contains 5,400 nucleotides. This is compared to 2,900 nucleotides of bacterial ribosome. And if we map all lengths of ribosomal RNA onto timeline, So we do see emergence of bacteria about 3.8 billions of years ago. We do see slight increase when we got emergence of eukaryotes up to here, but there is not so substantial increase. In fact, this increase does happen in both large and small subunits. However, it's not as pronounced for the small subunit as for the large subunit. And the critical event happens here once metazoans emerge. There is an enormous growth in ribosomal RNA at this point. So, and let me sort of say this. Does it always happen like this? Does always ribosomes grow and grow and grow as we saw so far during first 50 minutes or 40 minutes of my talk? No. There is a completely different example of opposite processes known in mitochondrial ribosomes. So, a couple of words. Mitochondria are endosymbiotic organelles that what used to be an alpha proteobacteria, now it's a little energy station that exists in every eukaryotic cell. So that's a late addition. And once an alpha proteobacteria became acquired into the eukaryotic cell, it lost a number of genes, and some of those genes got transferred into a nucleus, and at the same time, living inside of a eukaryotic cell, it just taken advantage of this fact that there is a neighboring nucleus, and it uses some of those gene products or proteins. So, such that the key point that the ribosomal mitochondrial ribosomes gave up a lot of their proteins from their mitochondrial gene into a nuclear gene. So, and apparently the smallest mitochondria contains only 5 genes. Human mitochondria contains about 15 genes. The largest mitochondria from a species called Reclinomonas americana contains about 100 genes. That's it. And only 67 of them are capable of protein coding. 30 other genes are ribosomal RNA and tRNA genes. And with that processes, the story changes completely. So what happens that once the genes, once the ribosomal proteins become redistributed between mitochondrial genome and nucleus, and mitochondria figures out how to take an advantage of additional gene products by exporting proteins from the nucleus. All of a sudden, it becomes depleted in ribosomal RNA, and it becomes substantially enriched in ribosomal proteins within mitochondria. And it goes, this is just quite recent publication from Science. This is a mitochondrial ribosome of a parasite, Trypanosome brucei. This is the only portion here shown in purple, what is a leftover of ribosomal RNA. The rest of it, for the small subunit here in yellow, for the large subunit here in blue, those are protein shells, remodeled protein shells. And surprisingly, the size of this ribosomal subunit is nearly doubled. This is compared to a bacterial ribosome shown here by this white contour line. So, and we've taken a look at the evolution and evolutionary pattern of all variety of mitochondrial ribosomes. And essentially, we try to reconcile all the available data. and map them onto secondary structures at the beginning. And then later on, we also rationalized everything in terms of three-dimensional structures. The problem is that there are no three-dimensional structures for all mitochondrial ribosomes. So we need to do this analysis using a combination of data coming from sequence, from secondary structures, and from three-dimensional structures. And what we saw that in many mitochondria, ribosomal RNA gets basically depleted, and the only region which remains is sort of shown here in this gray. That's a common core of mitochondrial ribosome. And it basically reduces down from 2,900 nucleotides to about 1,100 nucleotides. But there are some ribosomes, of course, that have more ribosomal RNA. That's basically just a limiting case. And of course, again, the same process correlates with a redistribution and repartition of mitoribosomal proteins. So here we mapped all these additions and deletions of mitochondrial mitoribosomal proteins for a variety of eukaryotic species. So we do have, of course, originally a mitochondrial ribosome inherited a full set of bacterial proteins shown here in black. And then as the evolutionary processes were going on, and of course this, the starting point for evolution of mitochondria is a particular alpha proteobacteria. The nature of this alpha proteobacteria is still being debatable. But what we do know is that this Reclinomonas americana, the species that I mentioned, it contains most bacterial-like ribosome, so there are no any deletions, there are no any accretions in terms of proteins. There is no any deletions, no any accretions for ribosomal RNA. But once we go towards this lineage, essentially we do see a number of deletions, all those black proteins shown in these red ovals with gray shadows. They're being lost, but new proteins are being accreted. And basically in Homo sapiens, we do see a substantial remodeling and enrichment of our mitochondrial ribosomes with additional proteins. And this slide is just a summary of some of the oldest mitoribosomal proteins that we do see. And what I just want to mention here, that those proteins are sort of localized within a couple of different regions. One is near the exit tunnel that interacts with inner membrane of mitochondria. The second cluster is here near the central protuberance. So I will stop at this point. Of course, there is many more to say. But let me conclude with the following. So I hope I was able to convince you that ribosomes are molecular fossils, and we can recover some information from ribosomes. Ribosomal RNA evolved by a process called an accretion. And a ribosomal common core is extended by eukaryotic extensions. Ribosomal extension segments reveal insertion fingerprints. The same fingerprints have been observed within the common core, and this is how we managed to reconstruct the entire model of ribosomal evolution. Ribosome gradually evolves by acquiring new functions needed to solve immediate problem. This is how new elements, those expansions and new proteins are being added on its surface. And in case of mitochondrial ribosomes, we do see some process which is totally different. We do see partial, to some extent, reversion. We do see a depletion in ribosomal RNA, and we do see an enhancement in ribosomal proteins. So mitochondrial ribosomal RNA loss destabilizes mitaribosomes, and the structural fragility of metastable intermediates makes them more receptive to accommodating new elements that stabilize the structure. So basically, we do see a completely opposite process, but it's not exactly the same. And the explanation for that is because in case of, and this is the most important message, in case of ribosomal RNA, when we talked about the evolution of a common core, the genetic code and the proteins have protein sequences were co-evolving with the evolution of ribosome. And ribosome was learning how to fold, proteins were learning how to fold, et cetera, et cetera. So there was a nice correlation. When we're talking about mitochondrial ribosomes, the genetic code is established. Not only is genetic code is established, those proteins, additional proteins, are available. They're available from the nucleus. And mitochondrial ribosomes, to some extent, they're just using this supply of readily available proteins and do this nice job of patching their own structures in order to survive and keep functioning. With that, I would like to thank all of you for this, for being here. I would like to thank Center for Origin of Life at Georgia Tech. I'd like to thank Lauren Williams and everyone from Georgia Tech who is working on these problems. And there is, of course, a special thanks to Professor Antonio Lazcano for inviting and hosting me here. And with that, I'm ready to answer your questions.
Thank you so much, Dr. Petrov, for this fascinating, beautifully illustrated sequence of events that led to the extant ribosome, or should I say to the extant ribosomes, considering the diversity that one can see in different structures from the different structures from the different biological models. Tenemos tiempo para preguntas y comentarios. Túnez Betabeles.
Dr. Petrov, thank you very much for your wonderful talk. I understand the model in which you say that ribosomes, the ribosomal RNA co-evolve with proteins, but how do you account for the proteins that interacted with the RNA, with ribosomal RNA before the origin and the establishment of the genetic code? I mean, because if I am not mistaken, you mentioned that in the phase 5, it's when the ribosome started to be in a coding phase. But that exact phase, you had already like a stage 3 of protein evolution, in folding evolution. So how do you explain, or what are your thoughts on the coding, origin of the genetic code and the involvement in the evolution of the protein interaction with the RNA? Thank you.
Thank you. This is a beautiful question. And so there are a couple of aspects that I should mention here. First of all, what we see right now, of course, those are the proteins, they're modern proteins, and all of them are products of the genetic code. So, and I want to make it very clear. So even those deeper regions located within the ribosome with a minimal amount of secondary structure or no secondary structure, they're still coded proteins. Furthermore, they are very conserved, most of them. So what we think about those regions, however, it's basically those new proteins sort of are the fossils of what used to be before in those regions. So we hypothesize that those regions have been replaced from non-coded unstructured proteins or some other polymers. And we have some ideas of what those polymers were, and that would be my second remark. And at some point, precisely around phase 5, which is the most interesting phase, the most transcendental phase of ribosomal evolution, when many events had happened at that point. When the code, basic code emerged and simplest faults have been formed, there was some replacements. So basically what we do see now, those tails, unstructured tails, they are fossilized records of what used to be within ribosome in the past. So now, to elaborate a little bit on what used to be there, and making a protein without a ribosome or with a minimal ribosome, it's still a difficult task. But it doesn't mean that some alternative version of a similar polymer cannot be formed. And there is an ongoing project going on at Georgia Tech, and it's led by Nick Hutt and Lauren Williams, and few people, including Moran, Frankel Pinter, and Jay Forsyth, have been working on that project. What they have been able to demonstrate that some slightly different polymers called deoxypeptides can be easily formed in the absence of ribosomes. And those deoxypeptides, which are just random mixtures of amino acids and oxyacids, you can easily form something with 30, 40 amino acids. This has been shown. And yes, we do not expect that at first glance that those proteins can form structured motifs, but they can easily interact with ribosomal RNA, and basically they can breach several ribosomal RNA helices, each other mimicking what those tails were doing. Okay, did I answer your question?
Let me just, for the sake of clarity, explain a little bit about what deoxypeptides are. Tradicionalmente pensamos correctamente en las proteínas como polipéptidos, la unión de distintos aminoácidos con enlace peptídico. Pero hay experimentos que se hicieron con el grupo que mencionó Anton Petrov y Ram Krishnamurti del Instituto Scripps en La Joya, en donde se demostró que, como en la Tierra primitiva, difícilmente puede uno pensar que había puros aminoácidos y puros aminoácidos proteínicos en charcos o en el medio ambiente, sino que también podemos sintetizar muy fácilmente hidroxiácidos o ácidos carboxílicos simultáneamente. Descubrieron que se pueden polimerizar abióticamente mezclas de estas distintas unidades, y el resultado van a ser polímeros que van a ser especialmente inestables después de varios ciclos de polimerización, precisamente en los lugares donde donde no tenemos aminoácidos sino otras alternativas, pero tienen propiedades muy interesantes, como este ligarse que no lo conocía yo al RNA en sitios muy específicos. El punto básico en esto es no pensar que en la Tierra primitiva habían únicamente los aminoácidos que estamos viendo hoy en día, o únicamente los polímeros de los aminoácidos que vemos hoy en día, o únicamente el RNA que vemos hoy en día, sino que había una diversidad química enorme en monómeros y en los polímeros.
Well, yeah, so, and your concluding remarks, if I understood those correctly, bring another sort of interesting point that ribosome and the genetic code itself was that filter.
Oh yes, absolutely.
That,
Right. Yeah.
That basically selected only those 20 amino acids that we now know as proteinaceous amino acids.
That's the basic idea. Hay un experimento por allí.
Buenas tardes. Me preguntas, ¿cuáles consecuencias serían en un futuro si se bioéticas o genéticas, si se modifica el ADN o si se clona una persona, un animal, o sea, en la alteración de la genética?
In humans, ribosomal RNAs, they're quite diverse. So basically, there are many copies. There are many copies of ribosomal RNA within human genomes. It's not just one copy, and those copies are located in multiple chromosomes. And what is interesting is that within the population, those ribosomal RNAs are diverse. So there is no single ribosomal RNA for humans. Furthermore, there is, even because there are multiple copies of ribosomal RNA within a single organism, there are some differences even within a single organism. So I guess this poses a big challenge for if we're talking about cloning and possible genetics. So there is still those exact differences in ribosomal RNAs. They are not very well understood. Why there is a heterogeneity of ribosomal RNA within us. So, but the implication is that there is a particular reason, and what it really means that very big accuracy has to be applied for this process.
Thank you. Sí, Don Omar, por favor. Perdón, sí.
Gracias. Sí, mi pregunta va relacionado con lo mismo, y esto precisamente, ¿qué cambios puede haber con las semillas transgénicas, con los plásticos? Gracias.
I would say that there are some, so if you're talking about sequence, a human ribosome is, as I mentioned, about 5,500 nucleotides. And at the level of sequence, all those 5,500 nucleotides, they look fairly the same. But when you look at the secondary structure, when you look at the three-dimensional structure, those nucleotides, the positions of those nucleotides, they mean completely different thing. So if one has a mutation or an error somewhere within those regions which are located not at the surface, those extensions, those mutations are not expected to be very lethal. In fact, those mutations they promote some changes within the ribosome. But there are certain regions within the ribosome that are extremely important and conserved. And during my talk, I mentioned that the central portions, the inner portions of the ribosome, tend to be much more conserved than the outer portions. So if one happened to have a mutation, and there are some nucleotides that are conserved such that everything alive has 100% conservation of nucleotides in certain locations. For example, PTC, peptide transfer center, and the decodic center, tend to be those invariants in sequence, not only in structure, but in sequence. So any single mutation in those regions would have to be lethal. And that's another point of concern here. So.
Don Omar, sí.
Good afternoon. You told us and you explained us the molecular arrangement of the ribosome, but I have here one particular question. You told us that the RNA, the ribosomal RNA co-evolved with the proteins, and the proteins gained complexity by folding, and you explained how the proteins were folding in your presentation. But I have a particular doubt. My doubt is how, or by what mechanisms, if it has already been discovered, can explain why the co-evolved development of the proteins and the ribosomal RNA. How, for example, me being a scientist, how can we explain, I don't know if it has been already discovered, but how can we explain this once that we understand that the proteins and the ribosomal RNA co-evolved separately and then arranged in a singular way, how can we, or by which mechanism we can explain this this arrangement, this singular arrangement between the proteins and the ribosomal RNA? And I don't know if I repeat, I don't know if the mechanisms has already been discovered, and I don't know it. But I would like you to explain and to clear this doubt out.
This is a beautiful question, and let me just mention first that we have this idea, and Lauren Williams sort of drafted it, and we got it elaborated. But the essence of this idea lies in the observation that and I will elaborate on that, the proteins and RNA, those are two essential biopolymers, and they are sort of, they complement each other. And if I want to extend this, I will argue further that those two types of proteins, so if you think of mutualism or symbiosis in biology, right? So you have different species, and they benefit from each other. They benefit from each other by living in a common ecological niche, sharing the same space, and then engaging in this symbiotic relationship. There is a beautiful variety of examples, but what I want to say is that in order to understand what's happening there within the ribosome, we just want to mention first that the RNA and protein are molecular symbionts at many levels, similarly to those biological symbionts that everyone knows about. And this is essential driving force, and I will list a few examples why this is the case. But this is essential driving force that responsible for coevolution of those two types of polymers and responsible for co-assembly of those two polymers. So in a brief, I can mention that first, ribosomal RNA and ribosomal proteins, they have a completely opposite fold. So if you think about it, what is RNA fold? So it has a backbone on the outside, it has bases on the inside, right? So, and then if you look at the proteins, what is an alpha helix? How it is organized? It has a backbone at the inside with the side chains going on the outside. So, and if you take any biopolymer or any polymer, in order to fold it, you have to undergo through those two scenarios. You would have to fold something or either outside in or inside out. And proteins and RNA, that those two limiting examples of those nearly perfect folds that nature has invented. That's the first thing. The second, when you talk about RNA assembly and the protein assembly, they're perfect polymers that engage into this assembly. Because if you think about hydrogen bonding, within a base pair, so we're talking about outside-in fold, base pairs are organized in a specific way that contains a number of donors, hydrogen bond donors, within purine and pyrimidine bases. Always equal the same amount of purine and pyrimidine acceptors, hydrogen bond acceptors. The same is for proteins. That basically you have the same number of hydrogen bonds, and this is how you make this perfect alpha helix. So being all this together, and I can elaborate more, but I try to make an argument that those two polymers, RNA and proteins, are those have been definitely refined, but as we know them today, they're perfect examples of molecular symbionts, because not only they different, they perfectly complement each other. What I mentioned, they complement each other in structure. But that has the next level of consequence. Of course, at the end, they complement each other in function. Where is a fold which is made by RNA or DNA ends up being perfect for storing all the information. The fold, so basically you have a very chemically inert backbone on the outside, and you're protecting all the information within the base pairs. So you have a perfect hard drive for storage of the information. On the opposite hand, you have this function with 20 amino acids forming all these globular structures with a side chain forming all these intricate pockets responsible for all kinds of catalytic activity. So we are going from two perfect polymers having this symbiotic relationship, molecular symbionts, but they are very similar in many cases, and yet they're different, and this results in difference in their structure. So, and sort of ribosome, it's sort of where bootstraps all these processes, such that at the end, by the time, by phase 6, we have those two things working together in a perfect way.
Si me permiten hacer un comentario al respecto, tradicionalmente la biología molecular trabajaba con los distintos tipos de moléculas o los distintos tipos de procesos en forma aislada. Había los expertos en ácidos nucleicos, los expertos en lípidos, los expertos en proteínas. Pero hemos llegado a un punto en donde ese reduccionismo ha sido realmente abandonado. Tenemos los instrumentos teóricos, los instrumentos empíricos, para ahora estar dándonos cuenta que en realidad los conjuntos de distintas moléculas están interaccionando de una manera tal que uno puede hablar de una coevolución general. Esa separación que hacíamos tradicionalmente ahora realmente se puede abandonar, porque si ustedes se ponen a pensar, no hay un solo proceso biológico en donde tengamos un solo tipo de molécula que no esté interaccionando con algo más. Piensen en el ciclo de Krebs, por ejemplo, tiene que haber claramente una coevolución de espacial, tiene que haber una coevolución funcional entre las distintas moléculas del ciclo. Y yo creo que el caso extremo que hemos visto hoy es una demostración efectivamente de cómo se ha llegado a un punto en el que podemos ir más allá de la descripción de un solo tipo de moléculas para verlo en dos tipos de casos. Lo cual me digo no solo por claridad para mí mismo, sino también porque hay una pregunta que yo le quisiera hacer a Anton Petrov, y es específicamente el RNA va a estar interaccionando con proteínas, con agua y con cofactores metálicos a lo largo de todo este proceso. Vamos a tener cofactores metálicos hidratados que pueden interaccionar con el esqueleto fosfodiestérico del RNA. Podemos tener cofactores metálicos que puedan ser parte del componente catalítico o estructural de las proteínas. ¿Ese tipo de modelaje ya lo estás haciendo o no?
Well, that's this is something that I was going to talk to you tomorrow.
Ah, okay. We're having dinner tomorrow. Okay,
yes, but before dinner. As a matter of fact, yes. There is an interesting sort of observation if we try to go a little bit deeper. So in my talk, I focused on those proteins that are universal, which means that that basically they do exist in both archaeal, bacterial, and eukaryotic domains. So those proteins, as much as I know, they do not have any metal factors in them. So, and therefore, for a while, we thought that maybe metals were not important, at least for the sake of ribosomal evolution. But then we started to look at some other proteins, and initially they did not look sort of universal. An example of those would be BL33, it's a bacterial protein BL33, and archaeal specific protein EL42. So they are zinc binding proteins with cysteines coordinating zinc. They are colocalized in the same space within archaeal and bacterial ribosome. But initially their genes looked like completely different until we discovered, well, we learned that at some point Tom Stites discovered that 15 years ago. But we noticed that those two proteins essentially is the same protein separated by a circular permutation. So this is why it's not detectable.
Okay.
So, and that was the first clue to look more at those zinc binding proteins that do exist within ribosome, but they are not universal. And then we started to notice some additional interesting phenomenon, and that's ongoing research, so we can talk about it tomorrow.
Lovely. Thank you. Sí, por favor.
Sí. Buenas tardes. Bueno, primero, gracias por la excelente plática que nos acaba de dar. Y quizá en la fisiología nos han enseñado que la mitocondria tiene procesos de hipertrofia e hiperplasia, pero ahorita estamos viendo que no es la mitocondria en sí, sino también el papel importante que juegan los ribosomas. Los ribosomas juegan este papel en mantener la síntesis proteica, y quizá nos explique la razón de por qué sujetos de más de 70 años pueden llegar a triplicar su fuerza o pueden seguir corriendo maratones a través del estímulo del trabajo de la actividad física que incide en lo que es el metabolismo energético y que se traduce a nivel mitocondrial. Gracias.
Well, so most of it is, there are some processes and there are some genetic diseases associated with mitochondria in general. There are few, it's a beginning of this new journey, and I can only make a small comment on it. The human mitochondrial ribosome have been discovered just 2 or 3 years ago by my collaborator Alexey Amunts, who is now in Sweden. He used to be working with Venki Ramakrishnan at Cambridge at the moment of discovery. And so it's essentially his work. And so there are some possibility of linking mitoribosomal structures to mitochondrial diseases, but at this point it's ongoing research, so I can only make this comment.
Don Omar, sí, por favor.
Thank you for a lovely talk. I just have a question. It was particularly interesting to learn about the changes in the mitochondrial ribosome. And I wonder if you know or can speculate about the possible changes in ribosomes from intracellular pathogens like Rickettsia, chlamydia, one of those.
So,
thank you.
Yes, thank you for bringing this up. And those organisms, those bugs, they basically represent an extremely interesting example, and it's basically, to me, it's almost a limiting case for mitoribosomal evolution. So those Brucei structures that I just showed, what basically happens that because of the genomic reduction in mitochondria, genomic erosion, all unnecessary ribosomal RNA is being replenished by proteins to an extreme, to such an extreme that proteins can intervene between RNA strands, basically those proteins can unwind ribosomal RNA, because when RNA is in a double helical form, basically there is a certain pitch and it takes a certain number of nucleotides to get from here to here. And imagine that these reduction processes would allow for RNA to go from here to here, completely sort of melted RNA. There is no canonical base pairing. It's just 2 strings of RNA going in the most straightforward fashion from point A to point B somewhere, and this whole cluster is stabilized by those additional proteins. So there are very interesting mechanisms, very interesting phenomena in RNA reductions. But at the same time, and I briefly made a comment on one of my slides, there are some critically functional regions, such that peptidyl transfer center in a large subunit and the decoding center in a small subunit. They always remain being rRNA made of rRNA. So, which basically still suggests at another level this symbiotic relationship. This is going back to one of the questions earlier. And this is another formulation of symbiotic relationship between protein and RNA. RNA makes all known proteins, and proteins make rRNA. So that's basically what we have now.
Or make the structure of RNA, if you wish.
Yes.
Okay. Bueno, ¿hay una pregunta más? Sí. Perdón, Martín.
Muchas gracias por una conferencia magnífica, Magnificent Conference. Hace unos meses tuvimos aquí a la doctora Ada Jonath y ella nos habló de su modelo del origen del ribosoma, y llegaba a un concepto de 2 moléculas de ARN muy similares que se habían emparejado una con otra para formar el núcleo, me parece que la peptidiltransferasa del ribosoma. ¿Ese modelo es compatible con el modelo de usted de evolución del ribosoma?
Yes, thank you for mentioning Ada Jonas' talk, and yes, so we are in a very good agreement with her model, and her model, the original model, was proposed in 2009 by her and her postdocs, and the whole idea of peptidyl transfer center being the oldest part of the ribosome, yes, it's basically the same. It's in agreement with this model. So in some respect, I would just have to say that the model that I described is an elaboration of ideas that stem from ideas of Ada Jonas and Sergei Steinberg on A minor interactions. It just, we provided a very, very detailed scenario for this process. Yes, the peptidyl transfer center is the oldest part of the ribosome, and this idea of first couple of helices that would comprise the peptidyl transfer center. In her model, it's those two pseudo-symmetric subunits. Yes, we are very, very, I mean, there are some discrepancies, but at the bulk, we are very consistent, we are in agreement.
Estaba viendo el teléfono celular no porque no estuviera prestando atención a la respuesta, sino porque llegó un comentario que me parece que puedo repetir para dar por concluida esta charla. Me pregunta una persona que no necesita ser mencionada, que por qué tanto énfasis en el Colegio Nacional en los trabajos de Ada Jonas, en los trabajos de Lauren Williams, en los trabajos de Anton Petrov sobre el ribosoma. Bueno, como se mencionó al principio y como han visto ustedes a lo largo de toda la charla, el ribosoma es claramente una estructura con una cantidad de información en términos evolutivos, estructurales, funcionales, absolutamente esenciales si queremos entender cómo surgió y cómo se desarrolló la vida en el planeta. Es absolutamente esencial si queremos entender la manera en que se forman las proteínas, sin las cuales la vida tal como la conocemos hoy en día no puede existir. Y como a veces la gente piensa que hay que dedicarse, esto era parte de la pregunta que me hacían por el teléfono, a veces piensan que solo hay que dedicarse a cosas que sirvan para algo. Déjenme comentarles que hoy en día, según las Naciones Unidas, la Organización Mundial de la Salud, uno de los grandes problemas que estamos enfrentando es el problema de la resistencia a los antibióticos. Es un problema tan severo que hay lugares en donde ya los médicos están pensando seriamente si se deben practicar cirugías o no ante el riesgo de que los pacientes sean infectados por algún microorganismo resistente a los antibióticos. Uno de los sitios de acción de muchos antibióticos son precisamente los ribosomas. Eso es algo que tenemos perfectamente claro. Entonces, en la medida que tengamos una idea más precisa, más detallada de cómo es la estructura del ribosoma, podremos entender cómo poder interaccionar con esas partes funcionales y evitar el problema de la resistencia. Hay casos, por ejemplo, en el que se metilan, se agrega un grupo metilo, un grupo C, carbono con 3 hidrógenos, se metilan sitios específicos en el ribosoma que impiden que el antibiótico pueda interaccionar con esa parte de esta estructura. De manera que se dan ustedes cuenta que la pregunta que me hacen, que viene de una persona que no voy a mencionar, es un reflejo de esa actitud simplista que debemos evitar a toda costa, que piensa que solamente se debe hacer un cierto tipo de ciencia que sirva, porque la frontera entre lo útil y lo básico es absolutamente incierta. La ciencia produce conocimiento y ese conocimiento se puede aplicar de manera inmediata o no se puede aplicar de manera inmediata. Y hay un elemento adicional en el que a mí me gusta insistir una y otra vez. ¿Para qué sirve conocer la evolución del ribosoma? No solo para entender estos modelos espléndidos como los que nos acaba de presentar hoy Anton Petrov, sino también sirve por el enorme placer intelectual el gozo del uso de nuestra inteligencia para comprender la naturaleza que nos rodea. Y creo que en la charla de hoy hemos tenido la buena suerte de tener un banquete de conocimientos básicos, multidisciplinarios, enfocados desde muchos puntos de vista, gracias a la espléndida conferencia que nos dio el doctor Anton Petrov. Anton, will always be your home. You will always be welcome here, and thank you so much for this extraordinary lecture. Ya para terminar, permítanme invitarlos a una mesa redonda que vamos a tener el próximo martes 17, después de las fiestas patrias, a las 6 de la tarde, aquí mismo, en donde vamos a conmemorar el nacimiento, el aniversario del nacimiento hace 250 años de Alexander von Humboldt. Alexander von Humboldt tenía un hermano, Wilhelm, ambos igualmente extraordinarios. Wilhelm von Humboldt fue el que insistió en que las universidades tenían que ser instituciones apoyadas por la sociedad y por el gobierno y que tenían que estar libres de presiones religiosas y políticas, que es algo que debemos tener presente constantemente. Y su hermano, Alexander von Humboldt, como todos ustedes saben, dejó la vida cómoda de un aristócrata alemán para venir al mundo americano y hacer unas descripciones absolutamente extraordinarias de la flora, de la fauna, de la orografía, de la geología de nuestra parte del mundo. Es una persona que amó a México profundamente y creo que es muy importante tener presente que el viaje de Humboldt a América se inscribe en este fenómeno de los viajes de exploración científica. Siempre pensamos en el capitán Cook, que murió en la Polinesia o en Australia o en algún lugar por allí, pero este es simplemente reflejo de esa expansión enorme que estaba sufriendo el conocimiento, la economía, la vida política europea y que finalmente en el caso de América produjo gentes tan extraordinarias como, o llevó a viajes tan extraordinarios como los viajes de Bonpland y de Humboldt, de Alexander von Humboldt. En la mesa redonda del martes, va a estar la doctora Edna Suárez, de la Facultad de Ciencias de la UNAM, que va a hablar del contexto en el que se dan estos viajes de exploración científica. Va a estar el doctor Jaime Labastida, que es uno de los grandes expertos en la biografía de Alexander von Humboldt, y va a estar Cédric Cerruti, un colega, un investigador francés, que ha trabajado mucho la obra de Bonpland, de manera que a nombre del doctor Luis Fernando Lara, del mío propio y del Colegio Nacional, los invitamos a que vean este otro aspecto del conocimiento de las ciencias naturales, que fueron los viajes de exploración del siglo XVIII, del siglo XIX. Y recordándoles que cuando Humboldt viene a México, se queda azorado de la riqueza tan extraordinaria que había en México, y en 1804 escribe que nunca había visto una sociedad tan refinada y con contrastes económicos y sociales tan dramáticos. Y desafortunadamente esa es una situación que seguimos padeciendo. Muchas gracias, buenas noches.